Excellent, thank you. Okay, yes, happy to be here with all of you today. I am a scientist in discovery toxicology at Bristol Myers Squibb San Diego, but speaking here with you today on behalf of the IQ MPS affiliate, which is a part of the broader IQ consortium. So I know that the slides will be made available for everyone, so I won't belabor in too much detail or spend too much time on our background information, but just a quick reminder for everyone about what IQ MPS is, bringing together multiple biopharma companies with the goal of creating external partnerships and collaborations, as well as establishing thought leadership and on MPS and complex in vitro model implementation in the pharmaceutical industry, creating ways that we can collaborate together and data share across companies, as well as creating opportunities for focused engagement between our industry partners and regulatory agencies. So a brief overview of what we are. We are now 27 member companies strong. And the way that we operate is that we have a steering committee that meets once monthly and is composed of 2 representatives from each member company. This is led by an executive committee, which you will see the current executive committee on the left-hand side of the slide. I am currently the past chair and will be cycling off for next year. So we run by elections, and we will have a new vice chair elected later this year that will be available for guiding the group. So as many of the speakers has mentioned today, we do have some challenges within the field with respect to our terminology, our definitions that we use. So just to orient everyone to how the IQ MPS affiliate has looked at the definitions and how we, think about things in all of our publications and workshop presentations and such. And I provided the references for where these definitions originated on the bottom of the slide, but we think of complex in vitro models as a broader, more umbrella term, as you can see. And then MPS is being, the more complex end of the spectrum, including things like bioprinted tissues and organ on a chip. But when we think about using them within the pharmaceutical industry, we really have to take a measured approach with respect to their qualification. And the points that I make today really originated out of an inaugural FDA IQ MPS workshop back in 2022, which the publication for that workshop is shown at the bottom of the slide. But we did have discussions around, what does it mean to be qualified? What does it mean to be validated in this particular space of complex in vitro models and MPS? And so many of the activities that we have had since then and the way that we talk about these models was really born out of those initial discussions. And we think of qualification and validation as separate activities, and qualification being a lower bar that you demonstrate that an assay or a model can be used reliably for specific interpretation. This most often occurs within a given laboratory or organization. So like, for example, within my laboratory here in San Diego at BMS. And it's often used for internal decision-making purposes, and we want to make sure that the demonstrated responses that we see within a model or an assay to known stimuli, align with what we expect. Validation, on the other hand, is considered a more formal, regulated or rigorous process. And this is really looking at, inter and intra-laboratory reproducibility, establishing data interpretation guidelines, and the like with more global reach. So think in terms of like an OECD test guideline as an example. However, just because a model is qualified doesn't mean that it can't lead to validation. It most certainly could, over time as you gain more confidence. But not all of the work that we do within pharmaceutical companies require formal validation of all the assays used. In some cases, qualification will suffice. But one of the things that we have recognized within the IQ MPS affiliate is that defining that context of use is really critical for both qualification as well as validation efforts. And you really must clearly and specifically articulate just how and when you want to use a model. So some of the examples that we utilize in this space, and is one of my favorites, is predicting skin toxicity. So in terms of how I might apply a model on my own programs within BMS, that's not quite specific enough. We need to take it a step further and utilize terminology such as determining the potential to cause skin corrosion or skin irritation. I use those in particular because those are formally validated OECD test guidelines. And ultimately, you really want to make sure you're asking the right question of the model that you are testing. Baseline characterization of the model can actually help you determine its suitability for different COUs. And so this is really important for, one of the speakers brought up today, immune-mediated DILI, and our paucity of models that can actually address this issue. Well, by implication of the name itself, you need to make sure you're utilizing models that have immune cells incorporated in it. So things like that, understanding the characterization of your model can actually tell you whether or not it is suitable for a particular context of use. At a fairly recent, back in 2022, workshop between IQ and FDA and other global regulators, the report, again, is referenced on the bottom of the slide. We really dug into qualification in the complex in vitro model space, and we utilized liver as an example and created 3 different categories of qualification: technical, organ specific, and then more context of use specific. Recognizing that, again, what you do and understand from your model and baseline characterization can inform what are the appropriate COUs to test based on that model's capabilities. And so I wanted to provide a couple of examples of how we've been thinking about this today. One being an example of qualification from the literature with respect to a transwell human ileum model consisting of primary intestinal epithelial cells and fibroblasts and ileal epithelial cells that was investigated for prediction of diarrhea as the context of use. So 39 compounds tested with and without clinical diarrhea effects, but all of which had no effect in 1 month traditional in vivo non-clinical tox studies. The researchers utilized TIR as the primary readout. And this is one thing that I do find that we have to talk about quite often within this field is the models may be increasingly more complex. But that doesn't always mean that the readout needs to be super complex. Sometimes you can use very simple readouts such as TIR, such as cytotoxicity to get the answer that you need. And what the researchers found is that adjusting the TIR values for clinical exposures actually improved the diarrhea prediction. So this is an example of qualification that was published, but not necessarily a formally validated assay. However, it still has utility within the pharmaceutical space. The OECD test guideline program, which other presenters have mentioned today, is a more rigorous validation process that has been applied to specific contexts of use. An example that I like to provide is reconstructed human epidermis, which previous presenters alluded to as well. This was one of the earliest complex in vitro models to be formally validated and largely driven by EU directives phasing out the use of animals in cosmetics testing. However, to date, there are at least 3 contexts of use that have been formally validated. The test guideline numbers are provided on the right there. And this approach has recently been applied for human reconstructed cornea models as well. One thing to note is the formal validation process, at least in what we have seen to date, does take several years of testing across multiple labs. But one of the things that is quite notable with this approach is that multiple models were available for each test guideline. So it's not just a single vendor or a single type of model that could be used within the test guideline. The test guideline offers flexibility. So depending upon what geographic region you may be in or accessibility to a model that you may have, you can utilize a number of models that have been validated within the test guideline to get the answer that you're looking for. So this is just really important as we start to think about, qualification versus validation in this space. A single model could be used for multiple COUs, but recognizing that for a single COU, you may be able to use multiple different models. And so I think we do need that type of flexibility within the pharmaceutical space because we are all geographically diverse and may have slightly different needs and questions depending upon the programs that we are running within our respective companies. So a brief summary, just to reiterate, defining that context of use is a critical first step to guide your qualification or validation activities, and that a single model could be used for multiple COUs or vice versa. A single COU could incorporate multiple models. Qualification and validation, we consider them as separate activities, but qualification can occur at multiple levels, both technical, organ specific, as well as context of use specific. And qualification can eventually lead to validation, but we don't always need fully validated like OECD test guideline type test protocols for the activities that we do in pharmaceutical testing. So if you do have any further questions, I've provided our contact information for the IQ NPS affiliate. The website QR code there will take you to many of the publications that I've referenced today. And you can also follow us on LinkedIn for any updates and notifications of our activities and workshops and new publications. Thank you, everyone.